Hello students, today's our topic is cell cycle. So what exactly cell cycle is? See, when a cell is formed, ultimately it is going to the next cell division. Usually it will be like that, okay? And the cell is undergoing so many changes to do the next division. So what are the changes which is taking place within a cell to do the next cell division? All these are collectively we can call as cell cycle. For example, the DNA is replicating and so many raw materials are forming in the cell for the cell division. The size of the cell is increasing. So these changes are periodic changes will be there, and we can predict what are the changes. Means it is a fixed type of changes, okay, and a fixed manner. There is a particular manner for the changes in a cell. So all these can be collectively called as the cell cycle, okay. And how many times a cell will divide? See, some kind of cells are continuously dividing. For example, meristematic tissue, it is undergoing continuous division. But in the case of a neuron in a brain, in the embryonic stage itself, the neuron is forming, and furthermore division will not be there for that particular cell. So that kind of cell also will be there. So how many times it should divide, that is already recorded in the genetics of that particular cell. Okay, so let us see what is a cell cycle. As I have told you, a cell is undergoing a periodic change to face the division, otherwise to undergo the division, and all these changes are known as what? Cell cycle. So generally we can see that the cell cycle has got two phases. What are they? The interphase and the M phase. What do you mean by interphase? It is a non-dividing phase. No division will be there in interphase. M phase, it is a dividing phase. Otherwise it is a mitotic phase. Otherwise we can call it as a multiplication phase. Here the division will be there. So in early times, the scientist has been believed that interphase is a resting phase. No changes is taking place in the interphase, like that it was believed. And afterwards it is discovered that it is not a resting phase. So many preparations are taking place, or so many changes are taking place in a cell to do the division. So in interphase, the cell is preparing itself to divide. So what are the materials it should require for the division? That is collecting in interphase. And as I have told you before, next to interphase, the M phase will be there. This is the division phase. In M phase, the cell will divide. Okay. So the interphase can be again divided into three more phases. The first interphase the first one is that first growth phase. First growth phase, otherwise we can call it as the G1 phase or the gap 1 phase. Okay. So the interphase is divided into three more phases. First one is first growth phase, G1 phase or gap phase. And the second division of the interphase is synthesis phase. And the third one is second growth phase, otherwise G2 phase or gap 2 phase. Okay. So the interphase is divided into three phases: first growth phase, synthesis phase, and the second growth phase. Okay. And after the interphase, the cell is undergoing what? The M phase. M phase has got two more divisions. Okay, M phase has got karyokinesis and cytokinesis. These are the two steps which includes in the M phase. Okay, karyokinesis means what? Karyon means nucleus. Kinesis means division. So karyokinesis means the division of nucleus is taking place in karyokinesis. Now afterwards, the cytoplasm is dividing, and that is known as cytokinesis. Karyokinesis is again divided into prophase, metaphase, anaphase, and telophase. All these things that we will go through the details of all these stages that we will discuss later. Today's our topic is the interphase. Okay. So in general, cell cycle has got two steps. Okay. And the first one is interphase and second is M phase. Interphase has got G1 phase, synthesis phase, and G2 phase. Okay. So let us see what are the changes which is taking place in first growth phase, otherwise the G1 phase. I'm going to represent it as a cycle, see, there is a very small phase. This is actually the division phase, otherwise we can call it as a M phase. This is a M phase, okay? M phase that we will discuss later. And now we are going to discuss about the interphase. The whole this space is representing the interphase or the non-divided phase. And as I told you before, this is the first stage of the interphase. This is the G1 phase, and this is the S phase, and this is the G2 phase, okay? G2 phase is comparatively a small phase when compared to the G1. Okay, like this. So this one is the M phase, otherwise this is the division phase. And this, all these things, all G1, S and G2, all these are the interphase. So firstly, we are going to discuss about the G1 phase. So after the division, a new cell will form, okay? Newly formed cell is entering into the G1 phase. A newly formed cell is entering into the G1 phase. What are the changes taking place in a newly formed cell in G1 phase? Firstly, see, you have to always keep in your mind this one, that all these phases are preparation for the next phase. Means G1 phase is for the S phase. The cell is getting prepared to face the S phase in G1, okay? And the cell is undergoing the changes in S phase to face the G2, okay? The cell is getting ready to face the next phase always. G1 phase, what are the changes taking place in G1 phase? That is making the cell ready to go to the S phase. S phase is making ready the cell to go to the G2 phase. Now what are the changes taking place in G2 phase is ultimately making the cell to do the N phase, otherwise division, okay? So in G1 phase, what are the changes which is taking place? G1 phase. So as I told you, see, S phase, in S phase the synthesis of DNA will take place, okay? The DNA will replicate here in S phase. To make the DNA, what I really need? So many enzymes it needs, okay? To make the DNA, it needs so many enzymes. Enzymes are basically what? Proteins. So that proteins are preparing in G1 phase. So the protein synthesis. The protein synthesis will get increased here in G1 phase, okay? And how the proteins are preparing? The proteins are preparing by means of RNA. 3 types of RNAs are there. By means of RNA, the protein can be synthesized. So that the RNA synthesis also taking place in G1 phase. RNA synthesis will increase in G1 phase, okay? And in in plant cell, the chloroplast will divide, and in animal cell, the mitochondria will divide. The division of mitochondria, and the chloroplast that you can see in plant cell. These two will be there in again G1 phase, okay? So what are the changes which is taking place in G1 phase is making ready the cell to go to the next phase, that is the S phase. In S phase, the multiplication of DNA is taking place, otherwise replication of DNA is taking place. To do that, the cell needs some kind of enzymes. Enzymes are basically proteins, and the protein synthesis will be high in G1 phase, okay? G1 phase is in G1 phase, the protein synthesis will be increased. To make protein, it need RNA. So that the RNA synthesis will also high in G1 phase, and mitochondria and chloroplast is also replicate in G1 phase. And when the DNA is replicating, to accommodate all these things, the cell should be enlarged its size, okay? The size of this cell should be increased. That is also taking place in G1 phase. Okay, the cytoplasm is increasing. The size of the cell is also increasing in G1 phase. So what are the changes? The protein synthesis is increasing, RNA synthesis will be increasing, and mitochondria and chloroplast will get divided, and the size of the cell is increasing. The cytoplasm is also increasing in G1 phase. Okay, and after that, after this G1 phase, the cell is normally going to the S phase, and some cells are not dividing further. Okay, so we have seen that after the division, new cells are forming. New cells are forming, and that new cell is entering into the G1. Newly formed cells are entering into the G1 phase. In this phase, the growth of the cell is taking place. After this G1, some cells are not going to the S phase. Okay, it is withdrawing from the cell cycle. Okay, it will go to the R phase, otherwise the rest phase. Have you understood? So normally after the division, the cell will be small and it should grow so that it is undergoing or it is entering into the G1 phase, and all these changes are taking place in G1, and afterwards normally the cell will go to the S phase. But in some cases, some cells are not going to divide furthermore. That kind of cells will be withdrawing from the cell cycle and it is entering into the R phase. Okay, so that is finished over here, and normal cells will go to the S phase again. Okay, so what are the changes which is taking place in S phase, otherwise synthesis phase? Synthesis phase, as I have told you before, the DNA synthesis is the DNA replication is the major point. Okay, the replication of DNA is taking place here in the S phase. And why the DNA should replicate? The DNA is doubling over here. So why? What is the need? See, actually the cell is going to divide. So when a cell is dividing, see, when a cell is dividing, for example, this particular cell is going to divide. When it is dividing, it is making two cells. Okay, to give the genetic material to both these cells, what exactly happening? How much DNA is here in this cell? It will double. Okay, and it is doubling, and one portion is equal portion is going to this cell and another equal portion is going to the next cell. For example, if it is a human cell, it has got 46 chromosomes. Okay, so the 46 chromosomes should go to this cell and it should go to this cell also, isn't it? So that the DNA should be double. Okay, one more set of chromosomes should be prepared. Okay, so to make another set of chromosome, it should need double DNA. Okay, so that the synthesis of DNA is taking place, and you have to always remember that the doubling of chromosome is not taking place in S phase. Only the doubling of DNA is taking place. The number of the chromosome will be equal. Okay, the number of the chromosome will be always equal, but the material DNA will be increasing. See, before going the division, the chromosome will be like this. Okay, the chromosome will be like this, and after the doubling of the DNA, it will be like this. The chromosome will be like this. Like this it will be. Okay, so here also one chromosome, here also only one chromosome. The DNA is doubled, but the chromosome is not going to be doubled. Okay, that is difference. So that is what is happening in S phase. Okay, synthesis phase. And DNA is increasing or DNA is doubling. And see, if it is an animal cell, it is going to the as I have told you, it will form the spindle fibers. Okay, the centrioles are going to divide. Okay, the division of the centriole is also going to be takes place here in the S phase. The centriole division is also going to be takes place. The centrioles are going to divide. That is also taking place in S phase. Okay, and the histone proteins are also going to make there in the S phase. And in some books you can see that the histone proteins are making there in the G1 phase, and in some books it is written in S phase the histone proteins are making. Both are correct because the lower division eukaryotes are making the histone proteins in the G1 phase also. Okay, the higher division of eukaryotes are making the histone proteins in S phase also. So what are the changes which are taking place in S phase? The DNA will get doubled. Okay, the synthesis of DNA will take place and the centrioles will be doubled, or the division of the centriole will be here, and the formation of histone proteins also is happening in S phase. And afterwards it is going to the third phase, otherwise G2 phase, or gap 2 phase, otherwise growth 2 phase. Comparatively it is a small phase when compared to the G1 phase. Here the cell is going to divide. Okay, here also see, the cell should make much more proteins. The division of the cell is also required so much kind of what proteins, or no, so many kinds of enzymes. Enzymes are basically proteins, no? So the protein synthesis will get continue here in G2 phase. The protein synthesis is continuing here in this protein synthesis. And G2 phase also the synthesis of the protein is continuing. And as I have told you, to make the proteins it need what? RNA. So the RNA formation is also continuing here. And another thing is that the ATP production. Okay, it is going to undergo what? Division, and it needs energy. So the ATP production is also increasing here in G2 phase. And afterwards, after this G2 phase, it is entering into the division phase. Okay. So once again, so what are the stages in interphase? Interphase means cell cycle is having two phases, interphase and M phase. Interphase is the non-dividing phase, M phase is the dividing phase. Okay. So interphase is having three stages. What are they? G1, S and G2. Okay. And G1 phase, in G1 phase, a newly formed cell is entering into the G1 phase, or the first growth phase. Here the protein synthesis is increasing, the RNA synthesis is increasing, and mitochondria and chloroplast is also going to divide in G1 phase, and G1 phase makes the cell ready to enter into the S phase. Normally it will go to the S phase. In some cases the grown cell, or the cell which the cell which entered in the G1 phase will withdraw the cell cycle and it is going to the rest phase. Otherwise it will move to the S phase. In S phase the synthesis of DNA is taking place, and the centriole division and the histone protein production is also taking place in the S phase. After that it will enter into the G2 phase. Here also the protein synthesis, RNA synthesis and ATP production will be there. And afterwards it is entering into the M phase. As I have told you before, M phase or the mitotic phase is having karyokinesis and cytokinesis. Means when a cell is undergoing into the M phase, firstly it is undergoing the nuclear division and then it is undergoing the cytoplasm division. So karyokinesis or the nuclear division has got prophase, metaphase, anaphase and telophase, and afterwards it is undergoing the cytokinesis. And the M phase that we are going to discuss in the next class. Thank you.